I don't know whether you have seen any of my other videos or not, but this one is an upgrade to an earlier one that I did, and I'm trying to explain the best way I know how to do why chromosome structure matters and why we need to get it right as well as we can. And I'm saying that when there's obvious evidence under a microscope to prove that chromosomes do not contain simple linear strands of DNA, and science works hard to say otherwise, when it's right there staring you in the face, it's time to do something about it. Now, science, at least regular scientists, not deep state science, but regular scientists live on a shoestring. They do. They have to be very careful about their funding, and they have to make sure that they stay within the lines. And that pays the bills in the lab, but it really hurts science because they can't go outside the box and look at things that are staring them right in the face, or if they do, they don't tell anybody about it. That is why it's so important for you to understand this video and let me know what you think about it. And if you have any suggestions, I leave my email at the end of it. Now, I know I keep ranting on and on about cancer and chromosome structure. The reason that I do is cancer, as far as we can tell, most of it is a genetic disease. Chromosomes break, they reform, they do all kinds of strange things that we need to understand what is going on. And in order to do that, you need to understand the mechanics of how that DNA is put together, how it operates, how it replicates itself, how it differentiates. You need to understand the mechanisms behind that. And if you don't understand the basic structure within the chromosome, you're never going to be able to figure that out. So without any further ado, let's get into the video. This video is an updated version of an earlier video uh, where you'll see a diagram similar to the one you see shown here. And this is a typical model for a cell and its nucleus, like you would see in cells like we have. This is a very typical model at the time that video was made, where you see the pink part at the upper part is the cell itself, which includes that purple region, the nucleus, and then within the nucleus are chromosomes, and then you'll notice they magnify that chromosome in this model, and you can see that one of the arms of the chromosome, which has been duplicated, one of the arms begins to unravel, or uncoiling, as it says here on the diagram, into a single DNA strand. So this model assumes that every chromosome, like this one, it contains but a single linear strand of DNA, all wrapped up and coiled up like a rubber band within a chromosome. What I'm saying here is that is not correct. That is not a good model for a chromosome. The unraveling is too simplistic. It does unravel, but it doesn't explain everything. And in fact, this model is so outdated, it just really needs to be removed from the premises. And let me explain why that is. This whole nucleus model cannot explain evidence, real evidence, of what I have seen under a light microscope. And let me show you what I'm talking about. Those little cutouts there that are green and yellow colored, those are what you find when I treat nuclei a certain way, a nucleus, as it says on the right. It's under 400x magnification using a UV light microscopy with dyes that stain DNA yellow. So those things that you're looking at and those little circles and everything are loaded with DNA. And they are not linear. They are circulars. So we need to be talking about circles of DNA instead of assuming it's just a big giant linear strand. I'm going to show you a new model that will help explain those grain photomicrographs up there. But first of all, to compare it with the old model, we need to understand something about chromosomes. So I'm not going to assume too much here. If you look at that chromosome in that old model, it has 4 arms to it. Let me show you. It's a duplicated chromosome. That's why it has 4 arms. There's the first arm, and there's the second one. They're duplicates of each other. And then there's the third, and there's the fourth, and they are duplicates of each other. But in the fourth one, you're seeing the unraveling taking place. What this shows here, it's a very strange looking model. It's an unduplicated chromosome, so it only has 2 arms, unlike the blue one that had 4. So there's the upper arm is in relationship to the blue chromosome, and there's the lower arm in relationship to that chromosome. And then the centromere is all those little speckled dots that you see. Now, I can go in a lot more detail about this later on, but this is just an introductory video. So I'm just trying to show you so you'll understand where this is going. And then finally, there's the telomere, which has a little hairpin on the end of it, and it's not shown on the blue chromosome. Now, if you magnify that section of the chromosome, that chromosome arm that is below the telomere, you see that you have a very strange looking object once you strip away everything but the DNA. And you have DNA circles. And so I'm going to count these circles for you. That one in the middle there, we'll call that circle number 1. And then that oval shape, which if you spread it out, it would also be a circle. That's number 2. The little circle is number 3. So there's 3 kinds of circles within that structure. And if you add all of those up, it amounts to 13 DNA circles. That's on the left side of that chromosome arm. If you go over to the right, there's another 13, which makes a total of 26 DNA circles. That big long oval is 1 DNA circle. It's huge. It's a huge circle, but it is a DNA circle. And then on the other side, on the right-hand side of that, is 13 more, which makes a total of 40 DNA circles. Now that lone circle that is by itself, that is part of the chromosome arm. That is a structural DNA circle to which all the other structures attach so they can be separated, replicated faithfully in mitosis. That circle there, I call it a complex replicon cluster. Those 13 circles are involved with replicating the DNA, as are the other circles on the other side. Now, if you were to take those circles and treat them as shown in the UV microscope photographs above, they will start to separate out, and they form 13 separated DNA circles. How does all of that happen? Well, that's the whole point of my research. Those green circles or yellow circles that you see in those microscope slides, they are much, much larger than what I'm showing here on the right. And they are more like a complex replicon cluster, and maybe even more complex than that. There is a hierarchical structure to these things that I explain in a lot more detail once you get a handle on what it is I'm talking about right here. Now this image, it's called a cell nucleus, highly diagrammatic, and it has these little compartments within it that look like everything's coiled up in there. And those things are called TADs. This is a mouthful, topologically associated domains. Okay, now what that means in plain English is that a chunk of DNA all wrapped up, it shows there, but it has its own compartment within the nucleus. So there's some organization there that is recognized by the scientific community. Now, exactly how that is organized, they are yet to say, but they are saying that there is some level of compartmentalization within a nucleus, like our nuclei. Now, I'm calling it a compartmentalized degenerate endosymbiont, and I don't really want to get into what that means right now, but we'll talk about that a little bit more later. Now, that's just a diagram like the other diagram. I'm going to show you reality, something that is not a diagram, it was found under a light microscope. We'll talk more about these two images later in the movie. So if mainstream science now assumes that the nucleus is composed of these irregularly shaped compartments, each one has a different color so that you can distinguish them with all this DNA raveled up in there. If we are to assume that that's correct, then let's take that old traditional model and paste this diagram right on there and assume that that is indeed the way it is, and I think there would be a consensus about that as well. So let's go back to that diagram and remove all those squiggly lines and just look at the focus on the compartments themselves. If you'll notice, they're very irregularly shaped. That's very similar to what an amoeboid shape, a protozoan. I want you to think about each one of these things as their own cells within the nucleus. And if that were the case, these would be called endo, which means within, symbionts, which means very similar to something like a mitochondria or a chloroplast. They are doing something within that nucleus that is unique to their functions that helps the cell, our kind of cells, to develop and become all different kinds of cells. And each one of these TADs or domains will have some function in doing that. They are coordinated and working together for the greater good of the whole. So let's just paste this diagram on top of that traditional diagram in place of the other one. Now, let's check out a new diagram. This one shows all of these domains, TADs, I call them endosymbionts, separating from each other. Now, what would cause something like that? Well, the cell is beginning to break up under some kind of external stress or internal stress, and these things are beginning to dissociate from one another. I'm going to slap that on that old diagram that we were looking at, see what that looks like, and then we're going to compare that to an actual photomicrograph. This is a photomicrograph that you saw. It's been cropped. You can see that these things are beginning to dissociate. They're beginning to separate from each other and spread out from the central core. And there's still a lot of organization here. But this is what's really going on. This is not some hypothesis. You're looking at it. It's actually happening. And so let's slap that on that diagram. Now we have another diagram of those amoeboid shapes, but they are surrounded by greenish looking circles of various sizes. Those circles are being ejected from those amoeboid shapes, which we will show with another photomicrograph coming up. So let's slap that onto that diagram and see where we go from there. Here's an actual photomicrograph of those circles escaping from that inner core. We're going to slap that on there also and see what that looks like. To me, it's pretty clear. So what we need to do is just eliminate everything about that old model and start with the cell and the nucleus.